Good evening, everyone. So we are the group 1 and 2, and we are tasked to report and conduct a test on tests for carbohydrates. So our objectives are to perform tests based on the production of purpural or purpura derivatives. Second is to perform tests based on reducing property of sugar. So these are the guide questions.
Number 1. What are the product form when each of the following carbohydrates is hydrolyzed? Number 1, sucrose. The hydrolysis of sucrose generates an equilamer mixture of fructose and glucose. Lactose. The hydrolysis of lactose are 2-beta-galactose. Maltose. When maltose are hydrolyzed, the bond between the two glucose molecules breaks down and maltose is converted into 2-glucose. Starch. The complete hydrolysis of starch yields the sugar glucose as commonly known as dextrose.
So why can't human digest cellulose? Human cannot digest cellulose because they lack the enzymes essential for breaking the beta-acetyl linkages. Unlike horses, cows, sheep, cattle, and etc., they also lack enzymes. However, the intestinal tract that contains bacteria that produces cellulose. In addition, termites also contains the same microorganism, which enables termites to use woods as their source of foods.
Number 3, explain the difference in the metabolism of fructose and glucose in the human body. Glucose is absorbed and transported directly to the body cells to fuel their metabolism. Eventually forms water and carbon dioxide to tricarboxylic acid or TIA cycle. It does not undergo any hepatic uptake, and in a state of excessive energy intake, another pathway is used to store it in the form of glycogen. And the third is to be converted to fatty acids and deposited in a fat tissues in the form of triglycerides. Fructose. Fructose has a low glycemic index or GI of only 23 compared to the glucose and used as the standard, which has a GI of 100. Ingestion is followed by rapid absorption, which leads to small postprandial, which rise in blood glucose. However, the liver is the main site of fructose metabolism, where fructose is converted to fructose 1 phosphate, thereby proving that it is not subject to phosphofructokinase regulation, which is the main rate limiting step in the metabolism of glucose.
Which of the following carbohydrate test is the appropriate test to detect the presence of sugar in urine? Explain why. Among the carbohydrate tests that were performed, sugar presence in a sample of urine can be detected by performing Benedict's test. Benedict's reagent is used to test for the presence of glucose in urine. The original color of Benedict's reagent is blue. It changes to green, yellow, orange, or red according to the concentration of glucose present in urine. Once a reducing sugar is detected in urine, further tests have to be undergone in order to discern which sugar is present.
So, test for the carbohydrates. So, in sucrose, in the Molis test, we already know that general test for carbohydrates. Then the positive color will be the purple. If sample were absent, if the sample were negative, then it absences of carbohydrate. So we already know that the sucrose has a positive. Then in Molish, then in the Benedict test, it is a specific test for the aldehyde. Then the positive appearance or positive test will be the red orange. orange. So it is a negative. So there's no presence of aldehyde. Then in Balfour test, it is a distinguish between the monosaccharide and disaccharide. If an sample were batted for 2 to 3 minutes, that means indicated for monosaccharide. And if after 10 minutes, if there's no changes, it is indicated as a disaccharide. So we all know that Balfour test for sucrose were positive, so it indicates as a monosaccharide, and that test color will be dark red. Then in the Bayal test, it is mm, it is test for pentoses and nucleotides. So the positive result will be blue green. Then it is a negative, so it is no presence of pentoses and nucleotides. Then in Siliwanov, it is a specific test for kentoses. Then the positive color will be bright red or cherry red or cherry. Then it is a positive, so it is a positive color, so the sucrose will be positive in Silivanov. So in iodine test, it is a negative. So next, please.
In glucose, the outcome is brown, brown, brown ink supernatant, and it is violet below, which is positive. And Molly's test. In Benedict, it is orange, which is positive. And in Barfold, it is dark red, which is positive also. No reaction happened. Also in Seliwano and in iodine, it is all negative.
So next is the fructose. For the Molish test, the result of the color in the Molish test is the color brown, and it is negative because it is not the right color. It is supposed to have a purple ring. And for the Benedict's test, the result of the color in the Benedict's test is the color orange, and it is positive because it reacts to the Benedict's reagent. Next one is the Barfoed's test. The result of the color in the Barfoed's test is the color dark red, and it is positive because it reacts to the Barfoed's reagent. Next is the Bial's test. For the result of the Bial's test, the color is violet, and it is negative because the right color is blue-green. For the Siliwanov's test, the result of the color of the Siliwanov's test is color cherry red or bright red, and it is positive because it reacts to the Siliwanov's reagent. And lastly, for the iodine's test, the color of the result is the red-brown, and it is negative because the color red-brown is equal to glycogen.
Next is the galactose. Galactose in the Molish test, a purple ring was formed, so it is a positive result because it is having a purple ring is a positive result of Molish. In Benedict's test, the galactose was color orange, so it is a positive result of Benedict's. At Barfoed's test, the galactose is color red, so it is also positive because the positive result for Barfoed's is dark red. In Bial's test, the galactose was color yellow, so it is a negative result because in Bial's test, it is supposed to be color blue. Next is a Seliwanov's test. The galactose in Seliwanov's test was yellow, so it is also negative because the positive result of Selivanov is cherry red or cherry red color. In the iodine's test, the galactose was color yellow, so it is also negative because the positive result of the iodine's test is the deep blue or purple.
So for the lactose, upon our observation, we saw a reddish violet ring of the junction of two liquids. That's why it is positive. So there is a presence of carbohydrates in lactose. Next is the Benedict test. So upon our observation, we observe a brick red precipitate, which is lactose is a reducing sugar. Next is the barfoids. On our observation, red scum is not seen. So that is why it is negative. So it is a disaccharide. Next is the bials and Siliwanov. Um, it is negative since we don't um since we don't since there is an absence of color or changes in the color in a period of time. Next is the iodine test. It is negative since um there is still no change in color and it indicates the absence of starch.
And the Molesh test in the ribose, it is positive because the Molesh test detects the carbohydrates present in the sample. So ribose has carbohydrates, so it is positive, and it is also have reducing sugar, so it is positive in Benedict's test. It is a monosaccharide, so it is positive in Barfoed. It is it has a presence of pentose, so it is positive in Bial, and it doesn't have it didn't react in the Saliwanov, so it is negative, and positive in iodine because it has a starch. In starch, it is positive in Molish because it has carbohydrates again, and it is negative in Benedict because it has no uh, reducing sugar. Barfoid because it is monosaccharide, so it is positive. While it has presence of pentose, so it is not negative. It has positive because it has presence of pentose. Negative, it didn't react into a Seliwanov, and it has starch, so it is positive in iodine test.
Distilled water. Um, in Molish test, the result of the color is brown, so it is negative. In Benedict's test, the color is light blue, so which means it is negative. In Barfold's test, there is no reactions even after 10 minutes, so it is negative. In Bial's test, the color is yellow-green, so it's positive. In Selivanov, there is no reaction, so it's negative. And lastly, in iodine's test, the color is yellow, so it's negative.
So in glycogen, since we were not able to perform it in the laboratory, we were tasked to research about it on the internet. So in glycogen, in Molish test, it produced a positive reaction. In Benedict's test, a positive reaction as well. In Barfoed's test, a positive reaction. In Bial's and Seliwanov's test, it produces a negative reaction, and in iodine, positive reaction.
So, so in conclusion, the development of a purple ring at the layer formed by the concentrated acid is a positive indicator for Malish test. In Benedict test, the Benedict reagent solution and reducing sugar are heated together. The solution changes its color to orange to red or brick red. This color is due to the presence of simple carbohydrates. Next, in Barfoed test, it is possible to distinguish between a reducing monosaccharide and a reducing disaccharide using Barfoed's reagent. A positive test is a dark red precipitate and is evidence of reducing monosaccharide.
In Siliwanov's test, the formation of the cherry red colored complex indicates a positive result, which means that the given sample contains ketosis. Result interpretation for Biles test is positive when there is formation of blue color, for example, ribose sugar. Negative Biles test, formation of any other color indicates negative test. Lastly, in iodine's test, deep blue or purple if there is a presence of starch, while red-brown in the presence of glycogen.
That is all. Thank you.